Everyone, and a very warm welcome to all distinguished guests, respected professors, colleagues, and students joining us today. It is my great honor the first Pakistan Physiological Society Research Committee webinar 2025, organized in collaboration with South Asian Association of Physiologists. Today's session will feature eminent experts sharing valuable
Assalamu alaikum, everyone. Thank you so much for joining from different parts of the world. First of all, we would like to invite the chief guest, Professor Rashid Mahmood, for his opening remarks and to make dua for Professor Inam. And Pakistan Physiological Society has just lost him, and we would like to continue his vision for research and education. Please, Professor Rashid Mahmood, the President, Pakistan Physiological Society.
Thank you very much, Professor Sabina Malik, for inviting me on this prestigious occasion. First, I congratulate you and your team, Professor Rehana and Professor Arif Malik, for this initiative. And we expect that this shall further flourish and shall continue. And you should transfer all of three, you will transfer your skills to our younger colleagues and younger physiologists. But before that, we proceed that I request everyone to recite Surah Fatiha one time and Surah Ikhlas 3 times. I shall recite and you recite with me, and we shall offer Fatiha for Professor Nam. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين بسم الله الرحمن الرحيم قل الله أحد الله الصمد لم يلد ولم يولد ولم يكن له كفوا أحد قل الله أحد الله الصمد لم يلد ولم يولد ولم يكن له كفوا أحد قل الله أحد الله الصمد لم يلد ولم يولد ولم يكن له كفوا أحد صدق الله العظيم. या अल्लाह जो तिलावत हमने की है इसका सवाब मरहूम की रूह को बख्श दे, उनकी मग़फ़िरत फरमा, उनके दर्जात बुलंद अता फरमा, उनकी कब्र में रोशनी अता फरमा, उनकी कब्र में नूर अता फरमा, उन्हें हर किस्म के अजाब, बरज़ख के अजाब, कब्र के अजाब, दोज़ख के अजाब, तमाम अजाब से बचा के रखो और उनके पसमान को हौसला और सब्र जमील अता फरमा। सदक़ल्लाहुल अज़ीम।
for your presence and support.
Amen.
Our first speaker for today is Professor Arif Malik, Director of School of Pain and Regenerative Medicine and Professor at the Institute of Molecular Biology and Biotechnology, University of Lahore, Pakistan. Professor Arif Malik has made remarkable contributions in the field of pain management, regenerative therapies, and cancer research. Today, he will speak on the topic organelle mislocalization and metabolic rewiring in cancer, the hidden role of compartmentalization. Sir, Professor Arif Malik, the floor
thank
is
you,
yours,
Dr. Inab, and thank you all the participants for these sessions. And I request Dr. Simina to upload my presentation to you. Dr. Simina, listen to me. I share my presentation with you on
okay,
WhatsApp.
Sir. Okay, sir. Let me open, please. Thank you.
Dr. Simina, are you view my presentation?
Yes, sir. I've received your presentation. I'm going to share it now. Just a second. Somebody has already started sharing. So now I'm sharing it. Here, sir. Please go ahead. Please go ahead, sir.
Hopefully, all of you look into the presentations.
Yes.
And today topic of my presentation is the organelle mislocalization and metabolic rewiring in cancer, cellular and metabolic perspective. Dr. Simran, next. Dr. Simran, are you listening to me? Yeah. Cell compartmentalization. The first part on my presentation is the cell compartmentalizations. And all of you know, cell compartmentalization refers to the way eukaryotic cells organize their internal environment into distinct membrane-bound and non-membrane-bound regions, each specialized for a specific biochemical and physiological processes. The purpose of this presentation is to understand a fundamental mechanism by which cancer cells adapt and thrive. This field of research aims to uncover how the abnormal positioning and function of different organelles of a cell, like mitochondria and the endoplasmic reticulum, physically drive the metabolic changes that fuel cancer's growth, survival, and resistance to treatment. Next, Dr. Smina. As I earlier said, just to understand the cancer metabolism, this type of research clarifies how cancer cells hijack normal cellular processes to support their rapid proliferations. It goes beyond the well-known barbaric effect to explain the underlying physical and structural changes that triggers metabolic shifts and identifying therapeutic. And the second part of this presentation is the identifying therapeutic targets by mapping the specific molecular pathways and the protein interactions that are disrupted by organelle mislocalization. Scientists can identify new targets for cancer drugs. The goal is to develop therapies that can reverse metabolic rewiring or specifically target cancer cells dependency on these altered pathways, leaving healthy cells unaffected. And the third part is explaining drug resistance. Many cancer treatments fail because tumor develop resistance. This field of study helps to explain how metabolic rewiring driven by organelle mislocalization contributes to this resistance, allowing researchers to design more effective combination therapies. Next, Dr. Singh. Here are a few features of a normal organelle localization. You all know mitochondria evenly distributed, localized near ATP demand regions. The next one is the nucleus, centrally positioned with the intact envelope, endoplasmic reticulum, perinuclear network linked to nuclear membrane. The next one is the Golgi apparatus, compact ribbon-like structure near nucleus, lysosome, perinuclear distributions, peroxisome, uniform cytoplasmic distribution, ribosomes attached to endoplasmic reticulum are free in cytosol. I just only repeat, I think all you know about this game of the cell or structure. I just only review the this normal cell organizations. The second one, next. The next causes of organelle localization in case of different type of diseases, pathologies, or specifically today we focus on cancer. First one, in this case, the cause is the organelle mislocalization refers to a pathological condition where cellular organelles lose their proper position distribution, or association within the cells. This disrupts the special organization needed for different biochemical or physiological processes and signaling. The second one is the cytoskeletal disruptions. Damage to microtubules, actin filaments, or intermediate filaments can impair organelle transport and anchoring. The next one is the motor proton defects, malfunction of kinesins, dyneins, or myosins affects directed organelle movement. Membrane tethering failures, loss of tethering proteins, Golgin's specifically leads to organelle drift. Pathogen interference, viruses and bacteria can hijack organelles positioning for replication. The last one is oncogenic transformation. Cancer cells often rewire trafficking to support proliferation and migration. The next one. This is the organelle mislocalization overview of a cell, specifically cancer-specific cells. Next one. Organelle mislocalization in cancer cells. Mitochondria clustering near nucleus, not in a central position, clustering near the nucleus, or laminopodia, right near the membrane. Nucleus enveloped disruption, micronuclei formations, endoplasmic reticulum. The fragmentation of endoplasmic reticulum, redistribution to cell peripheral sites. And the next one is the Golgi apparatus dispersed into mini stakes. And the lysosomes relocated to cell periphery, peroxisome loss or clustering, ribosomes redistribution to atypical sites. This is a comparative overview of a normal localization and mislocalization. And what happens in this type of mislocalization is the major metabolic dysfunctioning is that the VAR affect glutamine metabolism and ROS productions, oncogene transcription, DNA repair defects, lipid biosynthesis, endoplasmic stress adaptations, protein glycosylation, pro-tumor secretions, cathepsin release, specifically in cancer cells, which actually destroy the ECM degradation, extracellular membrane, and the altered lipid metabolism and ROS imbalance. And on the oncogene protein synthesis. Next. And two more things I added in my presentation, which mostly we pH and water, specifically designing or dealing with the patient cancer, are in our basic in vitro, in vivo research, the pH and the water potential, which have a very big role to designing or treatment of a cancer. The first one is the pH, which I enlisted here in a normal case in a mislocalized position, specifically in a cancer cell. pH. Next, Dr. Swin. Importance of different pH levels of organelles in cancer cells. Right here looks like a very small, and I asked all the present people in this presentation. I share this presentation because this is very too small to just read out after my presentation. I share the presentation. The next slide is the key tumor promoting mechanism of pH alterations. What happens in case of mislocalization in cancer cells? The first one is the metabolic advantage, means acidification of cytosol, and mitochondria enhances glycolysis and biosynthesis, feeding rapid proliferation. The second, invasion and metastasis, lysosomal and extracellular localization release ECM degrading enzymes, facilitating migration. immune invasion. Altered pH in Golgi and endoplasmic reticulum changes glycosylation, reducing immune recognition. The next one, the survival under stress. Endoplasmic reticulum and lysosomal pH shift support unfolded protein responses and autophagic adaptation. Sustained signaling, endosomal alkalization, extend growth factor, receptor, and activity. The last one, the genomic instability is a very important. Nuclear and mitochondrial pH shifts increases ROS mediated mutations. Next option. Again is the table which are two words are too small. Here I only read out the note. Water potential in cells is largely determined by solute potential and pressure potential, and for most mammalian cells is expressed in megapascals or as osmolarity. Intracellular water potential values are typical negative relative to pure water, with more negative values indicates higher osmotic concentrations. Next one. Role of water potential in cancer of saturation. Lower intracellular water potentials can cause an influx of water, leading to increased cell volume, which is often necessary for cell cycle progressions. This swelling can activate mechanistic signaling pathways like MAPK, P13K, AKT that drive production. Enhanced metabolic potential. Osmotic stress and altered water potential can recognize the cytoskeleton. The last one, cytoskeleton, making cells more deformable. This deformity allows cancer cells to squeeze through narrow spaces during invasion metastasis. Third one is support for metabolic rewiring. Abnormal water gradients influences ion channels and transporter activity, specifically sodium potassium ATPase aquaporins. This support nutrient influx, lactate efflux, and pH regulation, favorable condition for the Warbeck effect. The next one is the resistance to cell death. Hyperosmotic environment can trigger stress responses, specifically heat shock proteins and osmoprotectants that help tumor cells to survive against the oxidative stress, hypoxia, and chemotherapy. Altered water potential may suppress apoptotic volume decrease, a key step in programmed cell death. The last one, the tumor microenvironment remodeling. Changes in extracellular water potential influences interstitial fluid pressure, which reduces drug delivery efficacy, promote angiogenesis via VEGF signaling. And the last one, the facilitate lymphatic escape route of for metastasis. The next one is next metabolic rewiring. The third part of my presentation is the metabolic rewiring. This mislocalization and dysfunction of these organelles directly contribute to the cell's metabolic rewiring as indicated by the labels and arrows in the last previous cell figure. Increased glycolysis. This arrows promote, this is the depiction of the last figure of a cancer cells in which the mislocalization of the different organs are seen. The dysfunctional mitochondria cannot perform efficient oxidative phosphorylation, forcing the cell to rely on glycolysis, a less efficient but faster process that provides the building blocks for rapid growth. Altered lipid synthesis. dysfunctional energy production. And in summary, the last image visually demonstrates the fundamental principles that the physical disorganization of a cancer cell's internal structure, specifically the mislocalization and dysfunction of its organelles, is not just a side effect but a previous one. This is the Warburg effect. In normal cells, oxygen is essential for a highly efficient energy production process called oxidative phosphorylation, which occurs in the mitochondria. This pathway generates a large amount of ATP, about 32 molecules per glucose molecules. This is according to the model which already available in the literature. I am not in favor of this interpretation of a 32 ATP molecules. Hopefully, in next lectures, at this point, I'm not agree for 32 or 36 ATP molecules to produce in whole of the respiration process. When oxygen is absent, normal cells switch to anaerobic glycolysis, a much less efficient process. But cancer cells, however, continue to use a high rate of glycolysis even when oxygen is plentiful. They convert most of the glucose into lactate rather than fully oxidizing it in the mitochondria. This seems counterintuitive because glycolysis only produces about only 2 ATP molecules per glucose, but it provides a significant advantage for rapidly growing cancer cells. The next one is next. Why aerobic glycolysis benefits cancer cells? Fast ATP production. The first one is that biomass productions altered the microenvironment, survival in the hypoxic conditions. Next, Dr. Samina. Next one, Dr. Samina. The key to their survival lies into two main strategies. When the Krebs cycle is not fully functional in cancer cells, the cancer cell becomes highly dependent on the amino acid glutamine. They break down glutamine in a process called glutaminolysis. This pathway produces a key Krebs cycle intermediates, alpha ketoglutarate. The alpha ketoglutarate from glutaminolysis or glutamine metabolism refills the Krebs cycle. This process, known as anapullus, ensures that even a partial functional Krebs cycle has the necessary intermediate to continue running. Biomass production. The intermediate from this cycle are not primarily used for energy, but are instead siphoned off to produce the essential building blocks for a rapidly driving cell such as lipids, nucleic acid, and other amino acids. Cancer cells primarily rely on an aerobic glycolysis for their energy. This is a very fast but inefficient way of generating ATP from glucose, while the Krebs cycle is the most efficient way to generate energy. Cancer cells prioritize speed and the production of metabolic intermediates for growth. The Warburg effect allows them to generate enough ATP to survive. If the Krebs cycle is not fully functional, especially the last point in this, cancer cells use a combination of glutamine metabolism to replenish Krebs cycle, intermediates for biosynthesis, and aerobic glycolysis to quickly generate energy. This metabolic flexibility allows them to bypass the internal Krebs cycle and continue to grow uncontrollably. The next one. The current focus for therapeutic design, which currently in my lab is involved disrupting the glutamine metabolism. My lab targeting nowadays two type of pathways. The first one is the disrupting of glutamine metabolism by inhibiting the key enzyme with phytochemicals is a promising energy strategy to induce apoptosis in cancer cells. The rationale is to target the metabolic vulnerability that many cancer cells have developed, which is a heavy reliance on glutamine for their growth and survival. The targeted pathway, the process involves a two-pronged attack on the glutamine metabolism. The first one is the inhibition of the glutaminase. The first step of glutamine metabolism is the conversion of glutamine to glutamate, a reaction catalyzed by this enzyme, which is known by the name of a glutaminase. Inhibiting this enzyme effectively starves the cell of glutamic and its glutamate and its downstream products. This last one is the inhibition of glutamate dehydrogenase. This enzyme converts glutamate into alpha ketoglutarate, a key intermediate metabolite of the Krebs cycle. Inhibiting GDH prevents the replenishment of the Krebs cycle, further disrupting a cancer cell's ability to produce the building blocks of its need. The next one is mechanism of apoptosis inductions. By inhibiting these enzymes, these two enzymes, by inhibiting these enzymes and disrupting glutamine metabolism, cancer cells are forced into a state of metabolic stress that ultimately triggers apoptosis through several mechanisms. The first one is the glutathione depletion and oxidative stress. The second one is starvation for building blocks. And the last one is a disrupted mTOR signaling. Next, Dr. Sumina. The next one is direct enzyme targeting, evidence of inhibiting GLS and GDH enzymes activity or expressions from indirect modulators affect glutamine uptake, CMYC microRNAs, redox balance, or signaling that controls glutamine metabolism. Nowadays, I'm working upon the curcumin, moran from different plant sources, and AGCG and berberine and different type of flavonoids. First one is the quercetin, resveratrol, genistein, and sulforonil. Next one is Dr. Sumina. Mechanism by which phytochemicals can disrupt this type of glutamine metabolism. The first one is the transcriptional repressions, blocking glutamine uptake, direct enzyme inhibition, mitochondrial dysfunction, and metabolic flux, metabolic evidences, enzyme transporter expression and activity, bioenergetics, apoptosis markers, and functional rescue environments. Next one, Dr. Simina. The conclusion of my presentation is that many cancers depends on glutaminolysis, glutamine metabolism, from glutamine to glutamate and products alpha ketoglutarate and the TCA cycle by synthesis redox, blocking the pathway of GLS and GDH, both these enzymes, reduces the anaphylaxis, lower glutathione synthesis and is ROS, and impair biosynthesis, all of which sensitize cells to apoptotic death. Targeting GLS has been a validated strategy in clinical studies. GDH is also a pro-tumor bioenergetic regulator and an alternative target. this is a scheme of how I use my phytochemicals nowadays in this my lab. I am working upon both on these enzymes, GLS and the GDS inhibition, disrupted the glutamine metabolism, metabolic collapse in cancer cells. This is the last statement is actually my project. Last year I submitted in international organizations the name Infotech. My topic of research proposal for this competition, which actually worldwide, next generation interference with glutaminolysis, dual inhibition of glutaminase and glutamate dehydrogenase as a novel apoptotic strategy in a cancer. And for you people, Dr. Simina already know, and I declare here I win this world competition last Thursday. And this ceremony is hopefully on 17th September of 2025 in Istanbul University. And this research project is handed over to me by President Erdogan of Turkey.
Heartiest congratulations, sir. Very proud of you.
Thank you, Dr. Sumina. Thank you, Dr. Sumina. This is my whole story of my today. Currently working upon these two enzymes, and this is my topic of this research proposal which I submitted last year and win this year from whole of the world. Thank you. And if you have any kind of questions, please write in the chat box, and I hopefully help answer all the questions you raise.
Thank you very much, Professor Arif Malik, for your enlightening and thought-provoking presentation. Now, moving to our next speaker, Professor Samina Malik, Head of Department of Physiology at the University College of Medicine and Dentistry, University of Lahore, Pakistan, and the President of South Asian Association of Physiologists. She also serves as an advisor to the education mission of the Federation of Asian and Oceanian Physiological Societies, and as an advisory board member of the International Outreach Working Group, Center for Physiology Education, American Physiological Society. Her dedication to physiology research and international collaboration has been exemplary. Today, she will share her expertise on the topic oxidative stress markers for pre-diagnosis of breast cancer. Professor Sabina Malik, ma'am, we look forward to your presentation.
Good evening, everyone. Assalamu alaikum. So I'm very happy that we have participants from across the world, from Pakistan Physiological Society and SAAP. So as this topic goes, the paper regarding this topic has been published under supervision of Professor Arif Malik, my PhD supervisor, in Nature Scientific Reports, which you can see over here, and you can read the paper later. But I'll just make you go through the highlights. So you see that oxidative stress in the human body is one of the basic causes that generate the environmental conditions that eventually go to mutate certain genes that were otherwise tumor suppressive genes, and later on they become the genes that are responsible for the pathophysiology of breast cancer. And it is a fine balance of oxidative stress markers and their antioxidants both are present in the serum, and a fine balance has to be there. Whenever there is overexpression of oxidants and underexpression of antioxidants, then a situation can result like cancers and other diseases like Alzheimer's disease and heart diseases. So they play a role in the pathogenesis of breast cancer. You can see that on the right side, there are oxidants like MDA, interleukin, MMPs, tumor necrosis factor alpha, NO, INOS, heat shock proteins. On the left side, you can see SOD, catalase, GSH, and certain vitamins. So definitely we need to take these vitamins along with take steps regarding antioxidant and it is also called as anti-inflammatory diet, which is definitely going to help you. So building on, you can see that these are the values coming from the ROC curve to check the sensitivity and specificity of all the different oxidative stress markers that was measured in this project, in which there were 498 cases and 498 controls. The cases were all from suffering from breast cancer. You can see that this sensitivity percentage as well as specificity percentage was highest for 3 specific markers and sensitive markers, and these markers are starred on the left side. You can see in blue, the blue stars interleukin 1, and then heat shock protein 27, and you can also see interleukin 1, heat shock protein 27, and matrix metalloprotease 9. So these 3 markers were having the highest sensitivity and specificity, indicating their role in the prognosis and early diagnosis of breast cancer. So the simple test on which they were determined was ELISA, and the kit used was this one, ordered from USA. And simply the kit protocol was used to determine the levels. So what is the role of interleukin 1, heat shock protein 27, and matrix metalloprotease 9 in the breast cancer metastasis, these sensitive and specific markers? It is determined their interplay with the certain growth factors is one of the chief reasons to make them the highly sensitive and specific markers, like BCL2 and tumor growth factor beta and vascular endothelial growth factor. So these growth markers, the growth factors, they with these markers from the serum, oxidative stress markers from the serum, and they have a role to play in the pathophysiology of breast cancer. So especially matrix metalloprotease 9, it is found to be associated with this invasive ductal carcinoma. And you know that about 60% of the breast cancer histopathology is invasive ductal. So also it has been shown that advanced grades, that is grade 2 and grade 3 patients of breast cancer, they are frequently associated with high levels of matrix metalloprotease 9. So in case of Pakistan, I would say that as many histopathologists I have come to meet, none of them has ever witnessed grade 1 breast cancer, because patients, when they report to the doctors, they are already at the advanced grades they report with. So then there are many factors associated with it. One is the lack of awareness or health education, you can say. And there are other factors like the high cost of the screening procedures. So this study is basically based on a low cost and non-invasive approach by utilizing these very cheap ELISA-based biomarkers testing, and it will help you not only in the pre-diagnosis but also for the prognosis of breast cancer, and to determine the resistance to the chemotherapy that a person may face. So now we'll be talking about interleukin 1 and the cytoplasmic ERK pathway in breast cancer metastasis. So this interleukin 1 beta, which is one variant of it, with tumor growth beta and tumor growth factor beta and tumor necrosis factor alpha, it is involved in upregulation of another interleukin, that is interleukin 6, by activating this cytoplasmic ERK pathway, which is the extracellular signal regulated protein kinase 1 pathway. So the signals, they are picked up from the environment, and then they are brought to the cytoplasm of the cells. And in the cytoplasm prevails this ERK pathway, and then into the nucleus, the transcription takes place. So DNA is converted into RNA. You know that then translation will take place and a new protein will form. So associated obesity is also found, and this obesity is able to dysfunction the adipocytes. When the fat cells, they dysfunction, then there is release of interleukin 1 beta, and along with interleukin 6 and tumor necrosis factor alpha, it is able to cause the carcinogenesis. So this is the role of heat shock protein 27 in metastatic breast cancer, that is breast cancer with distant metastasis. So heat shock protein 27 basically stimulates the ubiquitination induced increase in cell proteins, and which is modified by SUMO2 or 3 to promote this heat shock protein B8 expression, which results into proliferation and metastasis in breast cancer, and it's been shown in mouse model and in vitro experiments. This heat shock protein B8, it and also inhibits apoptosis of cancer cells. So this way, it is able to promote the growth of the cancers. This is a postulated mechanism for interleukin 1. It is coming from my PhD thesis. Interleukin 1 is a diagnostic as well as a potentially therapeutic target, which may cause proliferation and is also involved in the migration and growth of the tumor cells, and especially in this type of breast cancer, that is invasive ductal carcinoma. So it also is involved in the metastasis. So you can see these pathways. So all these pathways are stimulated by this interleukin 1. It is stimulated by all these pathways, which are involved in cell proliferation and cell survival. So this is how it plays a role in the breast cancer pathogenesis. Here you can see how metalloprotease 9 is involved, which was also a highly sensitive and specific marker that we were talking about. So in a glioma pathway, that is glioma invasive signaling pathway, and here I have shown you the glioma metastasis pathway. So in this pathway, at the level of TIMP, you can see TIMP1, which is tissue inhibitor of metalloprotease 1. And what is it? It is a matrix metalloproteinase dependent regulator for growth as well as for apoptosis in different cell types. So its downregulation, you can see that downregulation of TIMP1, if it happens, then it causes upregulation of matrix metalloprotease 9. So then the potentially, that is why we can say that it is a potentially diagnostic marker and also therapeutic target. And mainly, we are talking about grade 2 cancers and grade 3 cancers, the high grade cancers. So it is specifically associated with high grade cancers. So next, we'll be talking about the pathway for the other highly sensitive marker, a highly specific marker, that is heat shock protein 27, the postulated mechanism. So you see over here, you can see HER2 new overexpression. So it can occur in aggressive breast cancer. And this is the heat shock factor 1, which it is able to stimulate. You can see that it is stimulating this factor. It is basically a transcription factor. So when it gets activated, then it is able to cause the overexpression of heat shock protein 27 as well. And this heat shock protein 27 further inhibits the apoptosis of cancer cells by blocking this mitochondrial pathways. So it is also associated with poor prognosis in case of breast cancer and resistance to chemotherapy as well. So in the end, I would like to conclude that raised levels of these 3 markers in the serum, they predictive value, not only for early diagnosis, but also we can say for pre-diagnosis. And even before there is clinical proof available, or even before there is some imaging sign available, like mammography or other images. So this is definitely a low cost and non-invasive approach, and it should be applied, I would say, to the community level. And it will be able to provide early intervention, and it can reduce dependence on expensive imaging in initial screening, which many people cannot afford and is usually offered at the age of 40, but the patients start suffering from breast cancer as early as age 18 very commonly. And it can improve the survival outcomes. And you know that the incidence or incidence of breast cancer in Pakistan is 1 in 9, and very similar in other South Asian countries. So we may be improve the condition of the patient, the prognosis or survival outcomes, especially in the low and middle income countries, because it is, it is very affordable and very cheap, and it can be afforded by all. So thank you so much, and we'll be open for any questions at the end. And I would request all the worthy participants to please type their questions in the chat box. Thank you. Over to Dr. Onam.
Thank you very much, Professor Samina Malik, for that highly insightful session and for highlighting new perspective in early breast cancer detection. Let us now proceed to our final speaker for today. Our third speaker is Professor Rehana Rahman, Professor of Biology and Director of Graduate Studies in Department of Biological and Biomedical Sciences at Aga Khan University, Karachi. She is a highly respected researcher, educator, and a mentor with an extensive body of work in reproductive health, nutrition, and metabolic disorder. Today, Professor Rehana Rahman will be speaking on the topic Vitamin D and Health, New Research, New Insights. Professor Rehana Rahman, ma'am, we are really eager to hear your valuable thoughts.
Bismillahirrahmanirrahim. Assalamu alaikum. Am I audible?
Wa alaikum salam. Perfect.
Thank you so much. Thank you very much for the very kind introduction. So I will be sharing few insights of the vitamin D research, which we have done and which we are doing. So what I will do is that I will tell us a little about the background of the study, what is the problem area, what is the work which we have done as far as the female subfertility is concerned, what is the work which is in progress regarding female subfertility and vitamin D deficiency. And similarly, we'll talk about the work done on the male fertility, subfertility, and what is the expected outcomes. So I need not to mention amongst the so many physiologists about the vitamin D absorption, vitamin D intake from the intestines, from the sunlight conversion, the formation of the 25-hydroxy cholecalciferol in the liver, and then the 125 cholecalciferol and 25-hydroxy cholecalciferol. You all are aware about it. So please allow me to spend some time on how we go for the research. The approach which we follow is the hypothetico-deductive cycle, which is that we identify a broad problem area, define the problem statement, and then develop a hypothesis, and then a design to give an answer to that hypothesis by collecting data and then analyzing it to have a final answer. So as you all know that infertility is a condition which is characterized by failure to establish a clinical pregnancy after 10 months of regular and unprotected relationship. And this infertility, there is an increase in the prevalence of infertility all over the world. But in Pakistan, the estimated prevalence of infertility has found to be 22%. Another part of our problem area is the prevalence of vitamin D deficiency in Pakistan, and it has been estimated that both the problems they have a well-known root. WHO has declared infertility as the 5th highest serious and global disability, and the prevalence of vitamin D deficiency, which is 53% in Pakistan, is also on an advancement. So, with the prevalence of infertility and the increase in the prevalence of vitamin D deficiency, which causes impairment of the normal male and female reproductive functions, spermatogenesis, oogenesis, disturbance of fertility is likely to occur. And with these two problems, we have hypothesized, and it has been published, that vitamin D deficiency, it causes increase in the insulin resistance, either by a reduction in the vitamin D receptors in peripheral tissues or by increase the pro-inflammatory cytokines. This causes hyperinsulinemia and androgen excess, which causes arrest of the antral follicle development, which is the one which is going to be to give the primary oocyte in the peritoneal fluid, which will be picked up by the sperm for the final fertilization. And this disturbance of this whole process leads to maybe a contributing factor to the polycystic ovary syndrome. So the vitamin D supplementation, we hypothesize that vitamin D supplementation can increase the endometrial thickness and can also lead to the maturity of the follicles. At the same time, by decreasing the anti-inflammatory cytokines, there will be an attempt to reduce the metabolic syndrome if the vitamin D treatment is given or if the vitamin D levels are adequate. So in a journal, which is in an article of Archives of Gynecology and Obstetrics, it was published that vitamin D has a role in insulin resistance. It has a role in ovarian dysfunction and oxidative stress and inflammation, as Dr. Samina has mentioned, that vitamin D acts as an antioxidant, and they have mentioned that these properties of vitamin D are because of its anti-inflammatory and antioxidative characteristics, by its role in the regulation of insulin secretion and tissue insulin sensitivity, and by regulation of the enzymes which are involved in ovarian hormone synthesis. So with the state-of-art lab available in the Department of Biological and Biomedical Sciences, Khan University, the multidisciplinary lab, we try our level best to do some research so that we find answers to the two problem areas which I have mentioned, infertility and vitamin D deficiency. So these are few articles which have been published on the basis of our research and on the basis of our different research projects. One of the work which is in the process of publication is the role of vitamin D receptors. And vitamin D receptors, as you all know, is located on the gene is located on the chromosome 12. It is a ligand-dependent transcription factor which belongs to the steroid hormone receptor family, and it has got the coding and the regions where the genetic variants can occur in the different mentioned positions. So what we found out was that there is a limited knowledge of the people about the genetic data which is available on the vitamin D receptor polymorphism and its association with female fertility. So we developed a hypothesis that a relationship exists between vitamin D receptor polymorphism and female infertility in Pakistan, and we explored the vitamin D receptor polymorphisms in infertile females and found out its association with female infertility. So these are all the methodological steps sample collection, DNA extraction, PCR, gel electrophoresis, and then designing the primers, and finally finding out what was the result. And we found out that the FOC1 had, on the basis of the regression analysis, that FOC1 had a causative effect on the progression of infertility, and the rest of the vitamin D receptor polymorphisms are one found not that significant. So our conclusion of this study was, among the 4 FOC1, BSM1, EPA, and TAC1, FOC1 SNP greatly increases the risk of infertility, while TAC1 and EPA1 genotypes protect from infertility, and BSM1 do not influence the risk of infertility. This is in the process of being published, and on the basis of this research, we have found out that the vitamin D receptor polymorphism should be checked, and the vitamin D receptor polymorphism should be corrected, or if not possible, the vitamin D correction therapy should be done before we opt for the costly treatment procedures for the infertility. This is the paper which was published in order to find out the interaction of single nucleotide polymorphism of vitamin D receptor and vitamin D binding protein, and its analysis was done by the bioinformatics, and we found that the plasma membrane estrogen receptor signaling enriched in deficient infertile females. We developed an animal model in which we used prepubertal female Sprague-Dawley rod, we left the control like that, and in the second group, we induced polycystic ovary syndrome, and in the third, we gave the vitamin D therapy, and you can see improvement in there was improvement in the uterine cavity appearance, as well as there was reduction in the number of the ovarian cysts, which have appeared because of the treatment given to induce the polycystic ovary syndrome. This has also been published, and on the basis of this animal, we are actually working on 2 grants. The one grant which is almost in the process of data analysis, and that is the efficacy of vitamin D supplementation and metformin compared to metformin alone in infertile females with polycystic ovary syndrome, a randomized open label trial, which has been funded by URC AKU grant. And the other one is efficacy of vitamin D supplementation in females with polycystic ovary syndrome, a randomized open label delayed start design, which is funded by Pakistan Science Foundation. And I will explain what are the differences and what are the similarities. So in the first, which is funded by our own AKU URC grant, we have recruited infertile females who are PCOS and who have vitamin D deficiency. Then we divided them into the intervention group, in which we gave vitamin D oral tablets as well as the metformin, and in the control group we just gave the metformin. Now we at 12 weeks we had a consultation with them, and after 24 weeks we performed transvaginal scan, as well as we looked at different their biomarkers and the changes which have appeared after the treatment with vitamin D, or if they have not occurred in the control group. The transvaginal scan, as I've already mentioned in the hypothesis slide, that we hypothesized that vitamin D improves the follicular size and endometrial thickness. So we will be aiming to see, is there any improvement in the endometrial thickness and the follicular size after the vitamin D therapy, and we are in the process of compiling those results. In the second trial, we are not taking the infertile PCOS, we are taking the young PCOS girls, and these girls are those who are actually or the nursing students or from any other any other entity of Khan University. And when they come to the family physician and they are diagnosed as a case of PCOS, we go for their vitamin D levels. And if their levels are less than 20 grams, then 20 nanogram per ml, we will recruit them in the as the intervention group or as the control group. Over here, the difference is that we will be giving not the oral vitamin D, but we will be giving the injectable vitamin D together with calcium. And in the control group, we are not only giving the metformin, but even PCOS care. And after 12 weeks, there will be crossover. So those girls who have received vitamin D will receive the standard PCOS, and those who have received standard PCOS receive the vitamin C. Whether the vitamin D treatment should be given to the females if they are giving the standard PCOS treatment, or it should be given after they have received the standard PCOS treatment. So at the same time, we are working on the male infertility, because we know that it causes the changes in the lipid peroxidation. It increases the oxidative stress, and on the basis of which we have published that vitamin D reduces lipid peroxidation and activity of reactive oxygen species, 4-hydroxy, 4-HNE, and nitration of tyrosine is done by it, and it uses the scavenging enzymes such as superoxide dismutase. These are the results on the basis of which we found out that if we divide the male subjects into two groups, those who have normal sperm parameters, which are total sperm count more than 39 million per cubic millimeter, total sperm motility more than 40%, and normal morphology of more than 4%, which is basically the criteria, strict Kruger criteria. It was found that there in the vitamin D was deficient in those male subjects who had altered sperm parameters, and these were the subjects who also had the oxidative stress products. HNE is compared over here. So at the same time, we are putting our efforts to investigate vitamin D hormone and oxidative correlates of male infertility to explore association of male factor infertility with semen inhibin. Previously, we had done our research on the serum levels, but now we will be looking at the oxidative stress markers in the semen and then compare them with the vitamin D levels, or correlate them with the vitamin D deficiency or sufficiency. We are also doing a culture experiment to evaluate the role of vitamin D in male oxidative stress, and to find out whether oxidative stress can be reduced by the administration of vitamin D in semen samples. So in the Dawn research briefing, we have published that vitamin D is crucial in the reproductive health on the basis of a study at AKU, as I have already mentioned. And with this, we hope that by being convinced first that vitamin D is required for the reproductive functions, in addition to the bone health, etc., a structured public health program could be launched to educate the masses, to correct their vitamins levels, to increase those kind of food which have high antioxidant as compared to the oxidants. And vitamin D can also increase the chances of normal conception and success after assisted reproductive techniques. So vitamin D screening should be done in or before giving treatment to any male or female component of a infertile couple before going to the very expensive tests. These are my references on the basis of which this presentation is made. And this is my book, which is going to be published, inshallah, by December 2025, which is Impact of Vitamin D on Health and Disease Beyond the Bones, on which we have taken the opinion and chapters from well-known gynecologists and those who are involved in the non-communicable diseases, etc., so that they all should convince the masses that vitamin D is important and vitamin D levels should be corrected, and we all should continue to move forward and see all those measures which can be done to improve health of our masses. Thank you very much.
Thank you very much, Professor Rihana Rahman, for sharing such detailed and updated knowledge on vitamin D and its implication for health. We have now
Thank
come
you.
to the interactive part of our webinar, where our audience will have the opportunity to ask questions from our esteemed speakers. To conduct this Q&A, I would like to hand over to Zimran, Punjab Counselor, Student Wing, Pakistan Physiological Society. Zimran, over to you.
Thank you for the introduction, Dr. Anab. I am Zimran Arif. I'm a second year MBBS student, and I'm the current Punjab counselor for Pakistan Physiological Society student chapter. And I will be mediating this session's Q&A session, starting off, of course, with Dr. Arif Malik. The question is asked by Abu Bakr Saud. Question being, in the context of the Warburg effect, could changes in mitochondrial positioning within the cytoskeleton directly influence the balance between aerobic glycolysis and oxidative metabolism in cancer cells? Dr. Arif Malik, are you there?
Hello, are you listening to me?
Yes. Did you hear the question?
Just to repeat the question.
Of course. The question by Abu Bakr Saud was, in the context of the Warburg effect, could changes in mitochondrial positioning within the cytoskeleton directly influence the balance between aerobic glycolysis and oxidative metabolism in cancer cells?
In real equation, the ROS production is enhanced by the mitochondrial metabolism, which is lethal for a cancer cell actually. In case of dislocalizations, the ROS production is enhanced.
Okay. Now we have another question from Dr. Ayaz Ahmed. I have put it in the chat so you could read it as well for your ease. The question is, what are the main challenges in translating this dual inhibition strategy,
Yeah,
GLS
I
and
already
GDH?
I write over the chat box. These are selection of the phytochemicals and testing them through computer drug designing, then on a rat model, then by the permission if you have a CRO license or from or your control trial from a WHO testing these phytochemicals. These are the challenges actually. Selection of phytochemicals, then computer drug designing, then rat models, animal models, or human models.
Thank you. Thank you so much for answering the question. We're moving on now to Dr. Samina Malik, our second speaker and chairman. Of course, Dr. Samina has already answered her questions in the chat box, but um, I have the questions here in case Dr. Samina would like to repeat them. Is that fine with you, Dr. Samina?
Yes, it's fine.
Okay, the first question is from Abu Bakr Saud. I've mentioned it in the chat box as well. Oxidative stress-induced exosomes are present in diabetes, inflammation, and breast cancer. How might targeting exosomal ROS pathways disrupt intracellular communication and halt tumor progression in comorbid patients?
Thank you, Zimran. So this is a great question, I would say, because it is tying together redox biology, exosome signaling, and the clinical problems of the patients with the diabetes or inflammation along with cancer, because this inflammation is common in all these conditions. So the compact plan or actionable scheme would be regarding how target exosomal ROS pathways, it could disrupt the tumor promotion and tumor promoting intercellular communication. So as I mentioned that first of all, exosomal ROS matters. It matters because cells under oxidative stress, if a patient is having diabetes, having inflammation, chronic inflammation, they release exosomes carrying ROS-related cargo. So ROS themselves, or you can say that ROS, they generate enzymes, oxidized proteins or lipids, redox-sensitive microRNAs, and mitochondrial fragments. So when these exosomes, they fuse with the cells of the recipient in the tumor microenvironment, then the cancer cells and the stromal fibroblasts, along with the macrophage endothelial cells, they transform the oxidative signals. And this will then reprogram the recipients to a pro-tumor state. So increased EMT, more migration, invasion, angiogenesis, immunosuppression, therapy resistance, as well as you can see metabolic rewiring will take place. So in comorbid patients like having diabetes, inflammation, and cancer, the systemic oxidative stress, it will amplify this exosome pro-tumor messaging. It will accelerate the progression and metastasis. Now, how can we disrupt the exosomal ROS pathway? We were saying that disrupting exosomal ROS pathway, it could help the tumor progression. So one of the ways could be reduced pro-metastatic reprogramming. This is one of the ways, and it will block the block transfer of ROS or ROS-inducing cargo prevents. It will prevent the activation of the signaling by different markers that will drive the EMT and MMP expression and motility. Another step could be restoring the anti-tumor immunity, that is lower exosome-driven oxidative stress or on immune cells like the T cells and natural killer cells. It can reverse the exhaustion or suppression. It will improve the immune surveillance and also the response to immunotherapy. Then we can say decreased angiogenesis and vascular permeability would be another factor. That is less ROS signaling to this endothelial cells. It will reduce the vascular endothelial growth factor and HIF-driven angiogenesis, and then vessel leakiness that otherwise supports metastasis. Similarly, improved therapy sensitivity, as I mentioned. So dampening the exosomal ROS signals, it can reduce antioxidant defense in the and this way it will sensitize therapy as well as radiotherapy. And finally, the limit systemic feed-forward loops. So interrupting the exosomal ROS can break the cycle in which the tumor and the comorbid tissues, especially the adipose tissue as well as the liver, chronically they will fuel each other with the damaging signals. Target strategy would be to limit exosome production release, to scavenge ROS or neutralize this oxidized cargo, to block the exosome uptake, or to target ROS sources inside the donor cells, or you can silence the pathogenic exosomal cargo, or combine all this with the standard therapies. That is, pair exosomal or antioxidant strategies with chemotherapy, targeted agents, or immune checkpoint inhibitors in order to improve the efficacy as well as it can also overcome the resistance. So I hope it explains. Thank you.
Thank you for the elaborate answer, Dr. Samina. We have a question from Dr. Ayaz Ahmed. In your study, when combining interleukin-1, MMP9, and HSP27, what was the aggregated sensitivity and specificity?
Yeah, thank you very much for your interest. But in this study, there were many markers, around 78 markers that were tested together, and the ROC curve was drawn together. So we did not study them separately. So for that, we will have to consider a new study in which we will keep only these three markers, and then we'll be able to calculate the aggregated sensitivity, because we performed on all those markers and created this diagram that you were able to see, in which individually we were able to show the sensitivity as well as the specificity and positive and negative prediction. So that is why it is not done segregatedly, so we cannot respond to this question. Thank you very
Thank you
much.
so much. Thank you. Another question from Uzma Ahmed. As it was mentioned in slides that the tumor markers in serum can be detected earlier than any imaging diagnosis, I am just curious if it is even prior to the mammogram diagnosis or detection.
Yes, thank you very much, Uzma. You are very right in your interpretation of what I was telling you, that if you go for mammography, they usually have this age limit, and they usually offer you, because it is not very common in Pakistan, not offered at many centers in each city. So that is why they have this age limit of 40 years, unless you have a very strong family history and very strong recommendation for that by the doctor. So, and that is also expensive. So yes, the answer is yes, that even before the development of breast, when mammography is not possible or not, important or not useful, even before the development of breast, in the serum, these markers can reveal a prediction. So you can predict with the help of these markers even before the breast is developed in a female. But in case of genetic markers, these are not genetic markers, but in case of the gene mutations in these are not the genetic markers, but hematological markers, oxidative stress markers. So there is no international criteria for them. So that is why we can take the edge of predetermining the onset of breast cancer in a girl even before her, adulthood, before the development of the breast. I hope it answers your question. Thank you.
The next question is from Nadia Mahasanul Islam. The question is, can you kindly tell the levels of interleukin-1, HSP27, and MMP9 for pre-diagnostic detection of breast cancer?
So Professor Nadia, thank you very much for joining from Bangladesh. And you can see in my paper in Nature, just type on Google Scholar Samina Malik, and then type Nature Scientific Reports and type breast cancer. So you will find this entire paper. So in that paper, you will be able to see the exact values of each and every marker. And thank you so much for showing this interest.
Thank you much for your time, Dr. Samina Malik. We are now moving on to Professor Anna. We have a question from Dr. Ayaz Ahmed. The question is, use a vitamin D antagonist for research, or how will you produce?
Okay, so actually, thank you very much for your very vitamin D deficiency will not be induced, but vitamin D, we will take the sample of the vitamin D deficient patients, and in vitamin D deficient patients, we will try to check how much is the oxidative stress. And in those samples, if the oxidative stress, those we will divide them into two groups, those which have the oxidative stress and those who do not exhibit the oxidative stress. Those which exhibit the oxidative stress by the administration of vitamin D, can there be any improvement? So this is only in the process of being finalized. But the hypothesis is that if we can improve the vitamin oxidative stress by giving vitamin D, we can safely say, we know that it is responsible, vitamin D deficiency is responsible for creating oxidative stress. But by this experimental study, we will be safe enough to say that vitamin D, by the cause-effect relationship, we'll find out the mechanism. Further, we'll explore the mechanism to say that vitamin D reduces the oxidative stress.
Thank you so much for your answer. Do we have any more questions for these speakers? Please mention in the chat box if you do.
This is Professor Sharmin from Bangladesh. Can you hear me?
Yes, Professor Sharmin, thank you so much for joining.
I have a thank you, ma'am. I have a very small query to Professor Rehana Rahman. I thank her for this very interesting presentation on vitamin D. Actually, one of my colleagues conducted one study to observe the relationship between obesity and parathyroid hormone. And it was observed that in obese women, parathyroid level was increased as because vitamin D was sequestered in the fat cells, that is adipocyte. So as because Professor Rihanna is doing so many researches on vitamin D, so my query is that as because in obese people, vitamin D is sequestered into the fat cells and so may give rise to secondary hyperparathyroidism. Is there any evidence that if there is intentional weight loss, vitamin D level will be increasing? I mean, the relationship between obesity and vitamin D, as because we know that the vitamin D is sequestered in the fat cells and also in obese people as because they don't go out usually. So vitamin D synthesis is decreased and also because there are some interleukin and leptin, which is secreted from fat cells. It inhibits vitamin D synthesis. So obesity and vitamin D is like doesn't go parallelly. So if we say that if there is weight loss, is there any improvement in vitamin D level in these people or women having?
Thank you very much, Professor Amin. Actually,
Yeah.
I did not go into the details of all the published papers, and in one of the papers which was published in 2025, we did mention that with increase in the obesity or with increase in BMI, there is an increased chances of vitamin D deficiency. But yes, we haven't conducted any study, any randomized control trial to see that whether the reduction in weight only can improve the vitamin D levels. But literature is full of researches which clearly prove that vitamin D deficiency is associated with obesity, or
obesity,
you can say that obesity, yes, as you very rightly mentioned, that obesity causes vitamin D deficiency. And in all the papers which I have discussed, we have found that obese women had low vitamin D levels. And in the lifestyle modification for reduction in weight should be undertaken at the same time while we are correcting the vitamin D deficiency. Thank you very much for
other hormone status. Thank you so much. Thank you so much.
My pleasure. My pleasure. Thank you.
Does anyone else have any more questions? You can come on the mic and ask any of these speakers.
I would also like to say that I have posted the link of my research paper that you can see in the chat box for further details.
Thank you so much. Okay, I'm assuming everyone has had their fair share of questions, so I'd like to call it a day. Q&A session. Thank you so much for being here. The e-certificates will be provided to everyone that attended and mentioned their emails and name, their identity in the chat boxes.
So thank you very much, Zimran, for moderating the Q&A session, and Dr. Anab for introducing all the speakers, and Dr. Maria for recording the video. And I'm grateful to all the speakers, Professor Arif Malik, my PhD supervisor, and Professor Rehana Rahman, a highly honored personality from Aga Khan University, the research head there, and being the members of this research committee of Pakistan Physiological Society for this great onset great achievement, the first webinar, and we would be continuing as required by the President of Pakistan Physiological Society, Professor Rashid, who joined us as the chief guest. And all the brothers and sisters in South Asia who are here with us. So we would be launching a basic and advanced research course, and it will be free of cost for the members of Pakistan Physiological Society, because the platform is Pakistan Physiological Society. And there will be only very minute or very little cost for the members from outside Pakistan. And if it is possible for them to manage the transfer, in that case only. And otherwise, the main purpose is to promote research from this platform, not only for the undergraduates, but also for the postgraduates and for the faculty members. So once again, I'm grateful to all the participants and all the speakers, along with the Pakistan Physiological Society and South Asian Association of Physiologists for this platform that was beautifully put together. And I hope that you found it useful. Thank you very much. Till we meet again. Bye-bye and
Thank you very
Allah
much.
Hafiz.
Thank you.
Thank you so much. Thank you so much for coming. Allah Hafiz. Thank you so much.
Allah Hafiz.
Thank you so much, Professor Ayub.
Welcome.